The evidence is becoming clearer and clearer and is pointing to the idea that SARS-CoV-2, aka the honey badger virus, is a human-made creation. Looks like it came from a lab. Hello everyone, this is Dr. Chris Martenson here with your daily update. It is March 4th, 2020. This is day 102. Wow, we broke into the triple digits. And I'm titling this one Lying Virologist and Self-Inflicted Wounds because I'm a little worried that it looks like this was a lab creation, and I'm going to get into some of the data behind that. We did a little of that on Friday. What was that? May 1st. But here we are. We're going to just finish up with a couple of pieces I didn't quite get to. So if you're a journalist who's investigating this, you're really going to want to listen in closely because I'm going to tell you the questions, or even just the question, that you should be asking to get to the bottom of this. But in due respect to the more than 3 digits now, we broke into the triple digits for our reporting on this. So guess what? Time to put on a little party hat. You know what? That's not quite enough. There we go. All right. That feels a lot better to me. Let's move on. Remember Friday we talked about this bombshell report in Newsweek, talking about how Dr. Fauci had backed controversial, and controversial is that's an understatement to me, Wuhan lab with millions of US dollars for risky coronavirus research. Okay. Very, very risky. And we talked about the risk of this stuff. This is called gain-of-function research, where they're effectively taking viruses that lack the ability to infect humans and experimenting on them to see if they can get them to infect more infectiously. Either they more easily transmissive or have more virulent, damaging effects on the body. So that gain-of-function research was ongoing because ostensibly the idea was, and I get it from an intellectual standpoint. First, we'd like to understand the mechanisms of how viruses do what they do. I get that, right? But the second point that they said was, well, you know, if one of these things did make this jump that we would sort of monkey with and predict and demonstrate could happen in a lab, then we would be able to figure out how to combat it more effectively. Here's the problem. I haven't found a single paper that talks about how we've gained any knowledge from this that would help us battle this particular virus that came out or any of the other ones. It's really, it seems to be the focus is mostly on intellectually, professionally, can we monkey with these viruses and do something with them? But at any rate, the story is now developing, this whole idea that it came from a lab. We see Mike Pompeo, U.S. Secretary of State, running around now saying enormous evidence the coronavirus came from a Chinese lab. Yeah, and I'm going to, the problem is that funding for that Chinese lab came from the U.S. So I'm not sure there's any clean shirts in this particular story. But to understand the science behind this, which is really important to understand, we have to take a little journey down into virology for a second. And really it's always been described that the way the COVID-19 virus gets into a cell is it binds to this ACE2 receptor. That's true, but it's really a 2-step process to gain entry into the cell. Binding that receptor is just the first step. So that's the first thing. Number one, it binds to the receptor. Now remember, we've already reported this could be ACE2 or it could be this CD147 receptor. ACE2 found distributed all through the tissues like we're talking about in this picture down here, like we see here. But CD147, that's actually in the, it's found all over the place as well, but in particular it's found in white blood cells and macrophages, things like that. So that's the first thing. You bind to the receptor. Here's the coronavirus. This is one of its S proteins, the spike proteins here. Here it is blowing up. That comes in and binds onto one of these ACE2 receptors. It's this protein subunit binds in and clicks onto it. But then a second thing has to happen. The S2 spike protein subunit, S2, the spike protein subunit, needs to be what's called proteolytically snipped or cleaved. And in this case, so there are various enzymes out there that are very good at cutting proteins, right? So that proteolytic cleaving requires another enzyme to come along. In this case, plasmin, they're looking at here. Plasmin, its normal job is to take fibrin in a clot and cut it up so that clot can dissolve. And when it cuts the fibrin, you get these other things called D-dimers, which you've heard me talking about as part of the clotting process. When they see really elevated D-dimer levels in people who have COVID-19, they say, oh my gosh, these people are in very bad shape. Once the D-dimer levels get up to a certain level, it means there's been lots of clotting, lots of cutting of those clots. But the other thing that plasmin can do now is it can actually make that snip in the S2 protein subunit of the spike protein, and that allows the RNA to fuse with the cell membrane so it can get into the cell. If that SNP doesn't happen, this thing will bind to the receptor, but then it can't get into the cell. So that clip, that proteolytic cut right there, really, really important. And plasmin is one of the things that could do the job. It is one of the protein enzymes out there, but there are many of them. And in fact, we need to talk now about something called furin, which is another one of these. It's a protein cutting protein. It's an enzyme that goes out and snips proteins, and its name is furin. Furin cuts proteins in strictly defined places. So it's a lot of fun to study if you're a virologist or other people who are working with proteins, because you can insert a sequence of amino acids, and furin will cut very, very defined places in there always. Some of these proteolytic enzymes, they go out and they cut all over the place. But furin's very, very strict in what it does. And it cuts after this sequence, RXXR, and that is arginine. The R stands for arginine. That's an amino acid. XX, that could be any amino acid, and then another arginine. So if you see this pattern, RXXR, then that's a sequence that furin is going to come along and make a cut there. Moreover, however, you go even further, if arginine is also in the second or third place, so here it is in the second place right here, here it is in the third place. So if you have this RRXR or RXRR, then the cleavage efficiency is significantly increased, okay? And why is this important? Well, this is the smoking gun in this whole SARS-CoV-2 story here right now. This is the one thing that we need to focus on, the one thing only if we're going to be chasing this down. So this person, by the way, in this medium.com article by Yuri Dagan here, I'm going to be drawing on a lot. It's just a fantastic, amazing article that just lays out study after study, piece of data after piece of data, and explains it all in very simple, beautiful terms. So Yuri wrote here, therefore, the appearance of a new furin cleavage site, which was noticed in SARS-CoV-2, there's a brand new site, was noticed immediately, as none of the closest or even distant relatives of SARS-CoV-2 have such a site. Those coronaviruses that do only share 40% of their genome with CoV-2. So one of the things we're trying to do here when you're trying to figure out, hey, was this natural? Was it man-made? Where did it come from? You always have to go with the sequence, how close the sequence of this particular virus is compared to its closest relatives. In an ideal situation, if it did come from nature, we would find a natural virus that has exactly the same sequence as the one that was found in patient 0. Now, remember, patient 0 probably was in China and probably was there probably November or maybe October, but sometime back there. And that was the first exposure, that patient 0. After that, the virus begins to mutate a little bit. And these are little tiny single nucleotide polymorphisms, the SNPs. We talked about those, right? So over time, it will start to mutate a little bit. Most of those mutations mean nothing. They confer nothing new to the virus except it has a slightly different sequence from the one that came before it. And this is how they're able to track who got it from who, where, when, and all of that stuff, right? So that's why in a perfect world, if this particular virus did come out of the natural world, in that perfect world, we would know patient 0, and we would also have a natural reservoir, and we would say, hey, look, those are identical. Those are the same sequences. And then the case would be closed. All right, it was found that all spike with a SARS-CoV-2 spike sequence homology greater than 40% did not have a furin cleavage site. So this is including this BAT-CoV, the rat TG13, and SARS Classic here, with sequence identity as 97.4% rat TG13. That is the closest one. SARS-CoV 78.6% homology sequence identity there in the spike proteins. But they found this furin cleavage site RRAR in SARS-CoV-2. That's RRAR. That A, you can think of that as an X, because that's this right here, RRXR, right there, okay? So a cleavage efficiency is significantly increased. And remember, you need to cleave before you can actually infect. Two-stage infection process. Step 1, bind to the receptor. Step 2, get cleaved in a specific spot on your protein subunit, allowing this conformational change take place, and then next thing you know, the viral RNA can gain entry into the cell because of that fusion process. All right, the furin cleavage site RRAR in SARS-CoV-2 is unique in its family. That's an important sentence right there, is unique in its family. Rendering by its unique insert of PRRA, the furin cleavage site of SARS-CoV-2 is unlikely to have evolved from MERS, HCOV, HKU1, and so on. From the currently available sequences and databases, it is difficult for us to find the source. And here the scientists are saying perhaps there are still many evolutionary intermediate sequences waiting to be discovered, which let me decode that sentence for you. They're throwing their hands up going, there has to be some magic that happens between the closest relative we know about to SARS-CoV-2 and SARS-CoV-2 itself. There's some magic that happens between there and there. All right, so this is interesting because now we're going to tie all this together and then get back into the sleuthing a little bit. Because what's interesting, we looked at those clinical risk factors and the clinical observations that said, wow, all these people with, right here, hypertension, with diabetes, people with coronary heart disease, cerebrovascular illness, chronic obstructive pulmonary disease, kidney dysfunction, these people all have worse clinical outcomes when infected with SARS-CoV-2 for unknown reasons. And by the way, they could have also put obesity in here, and that ties into this story very, very tightly too. The purpose of this review is to summarize the evidence for the existence of elevated plasmin in COVID-19 patients. So plasmin is one of those proteolytic enzymes that comes out and clips proteins, right? We talked about that, and it clips fibrin and creates those D-dimers. All right. Plasmin and other proteases may cleave a newly inserted furin site in the S protein of SARS-CoV-2 extracellularly, which increases its infectivity and virulence. Hyperfibrinolysis associated with plasmin leads to elevated D-dimer in severe patients. The plasminogen system may prove a promising therapeutic target for combating COVID-19. So they've already noted clinically that this extra cleavage site is conferring, ties in very tightly with these particular outcomes in the clinic. And by the way, people who have these various conditions tend to have elevated levels of plasmin and plasminogen in their blood. So this is beginning to tie together pretty nicely. Extracellular cleavage of virus envelope fusion glycoproteins by host cellular proteases is a prerequisite for the infectivity of respiratory viruses. And by the way, we've known that for a long time, decades now, okay? The presence of a polybasic cleavage site. It's polybasic, so the arginine is a basic amino acid, so that RRXR, that's what they mean by polybasic, many basic pieces together, RRXR. That can be cleaved by furin-like proteases is a signature of several highly pathogenic avian influenza viruses. Oh, and by the way, have humans been monkeying around with avian influenza viruses inserting these polybasic cleavage sites to experiment? Yes, we have. Similarly, the S protein of SARS-CoV-2 harbors a furin cleavage site at the S1, S2 boundary. That's the subunit 1, subunit 2 of the spike protein. And we talked about that just a second ago. The almost ubiquitous and diverse expression of furin-like proteases could lead to increasing SARS-CoV-2 cell and tissue tropism and transmissibility and enhance its pathogenicity. All right, so the clinicians are starting to weigh in and they're starting to look at this. So let's look back at COVID-19 because this is the smoking gun part of the story here. So let's look here. By the way, sorry, this is kind of small here, but this is a table pulled from a study that was looking at SARS-CoV-2 here at the top, right? And here are all these other coronaviruses that it could be related to, okay? And here's its identity. Here's how close its spike protein is. So SARS-CoV-2 is 100% identical with itself. So that's a good thing to find, right? Its next nearest relative is the rat TG13. It has 97.4% similarity in the spike protein, but this is its furin score, okay, which is anything over 0.5, it says, means that it's furin cleavable. So SARS-CoV-2 has a 0.62. Its nearest relative on its family tree, if we want to look at it that way, only has a score of 0.15. And then down here at 80%, you have one with 0.17, Here's one at 76%. So we're getting really far down the family tree. 0.117. Oh my gosh, you jump all the way down to only 37% identity here with the HKU5. And here the furin score jumps back up to 0.697. Here's MERS itself at 0.563, but only a 35% identity. Rat Cove here, 0.879 on the furin score, but only 36% identity. So here's the thing. No nearby relative. Whoops. God, why doesn't that pick that? There we go. No nearby relative has a furin score that's useful. These are all not, they don't have furin cleavage sites. These do. These all have really tasty furin cleavage sites, but none of them are more than 40%. In fact, 37 is the highest. So none of them are more than 38% similar to the one in question, COVID-19. So that is a big, big deal. It means that this thing somehow magically all of a sudden got itself a furin cleavage site, which we can't see anywhere on this family tree. This is a little hard to interpret. Here's another way I saw it put. I like looking at it this way. And what we're looking at here is the pink means that let me back up here. These are all coronaviruses, and this is a coronavirus family tree right here. And you could think of each one of these as you come out this tree. These two would be very closely related to each other. These two little things right here, I can't even see them, so if it's blurry for you, don't worry about it. But you'd come all the way out here, and this is a big tree right here. And then we have to jump all the way over, and there's another tree right there, and you come all the way out here to another big family tree, and then here. You can think of each of these as like grandparents. There's great-grandparents, great-great-grandparents, great-great-great. This is really, really far genetically, getting further and further apart genetically. All right. So this is a genetic wheel, and it shows how close these things are. So these are all closer to each other, and by the time you're over here, you're really far away from these, and so on. The pink ones, anything in this pink part here, these have furin cleavage sites in their sequence. And the ones in yellow have no furin cleavage sites anywhere in their sequence, okay? And here's the punchline to this. This is SARS-CoV-2 right here, and here it is with its own little tiny wedge way out in the middle of a whole lot of yellow. It doesn't have anybody related to it anywhere close in its family tree that has this furin-specific cleavage site. It's just parked all out there all by itself, and you have to go back many generations, many generations. You have to come all the way back to here to come down this spoke of the wheel in order to find anything close to it. So that's quite an oddity. Normally, the way these things would go is that you would find that everybody's pretty closely related, and if you wanted to see how this got its furin cleavage site, you would just trace back and you would come back down another one. Like you could do that here, like up and down like that, right? We're still in the pink part of this wheel. So at any rate, real mystery, just presented by these people like, yeah, look, there it is, lurking all by itself way out there, SARS-CoV-2. And let's see, where do I want to go with this next? Yeah, okay, I guess we'll go this way. Yeah, we'll do this. All right, so this is comparing the actual genetic sequences. Here's the rat TG, not that one. Let's get the other one out. Rat TG13. Here's SARS-CoV-2, right? And here's pangolin, the M789. That's one of the closest relatives. So you've heard big prominent virologists, Michael Osterholm and all these people who were in that Nature paper. I'm going to pull back up again, all saying, looks like it came out of a bat, but maybe it had some time in a pangolin. One thing I want to have you look at here is just how close these all are, except here's that PRRA sequence right here. And it exists in SARS-CoV-2, but it's not just, this isn't a mutation. It is completely missing. That's what these dots mean. It's completely missing. So now we have to talk about something that's really important that is part of this smoking gun story. The difference between a mutation and an insert. So this is the genetic string of RNA going along, and this is actually from the pangolin right here that I pulled out. And this is a mutation right here, where instead of a T, there's a G here. And because of that, you will get a different amino acid. Why does this pick so badly? There. Because of this, you will get a different amino acid out of this. So these are read in threes. So this is the way cellular machinery works. These 3 RNA chunks will be read by a ribosome. It'll come along. It'll look at all 3 of those together, CAA, and it'll give you an amino acid because CAA confers a specific amino acid. And here's another one. ACT is another 3-letter combo that gives and codes for a very specific amino acid. It reads along. AAT gives you another one. TCA and all of that. And so we're just going along like this. They're exactly identical, right? They're exactly identical here. In this sequence right here, I'm reading this sequence right here until you get here. And then all of a sudden this G is different from that T right there. Okay. So that T and that G are different. And that's what an insert is. These mutations, sort of mutation is, these mutations are fairly common. They happen pretty often. What doesn't happen all that often is you get this giant insert where there are 12 new base pairs in here. Nucleotides in here, not base pairs. Sorry, I get my DNA mixed up. This is a single-stranded RNA. So this is just 12 nucleotides. CCT, GCC, CGG, CGA. That gives us our PRRA. This sequence right here gives us that polybasic furin cleavage site. All right. And here's what's interesting. Remember we talked about, and again, this is a note to journalists. You just want to remember this PRRA. You just want to get this. We talked about this article. It came out here, the proximal origin of SARS-CoV-2. We had Robert Gary in here. We had Lipkin. We had Anderson here. And they came out with this, big, giant study that came out in Nature or a letter that they wrote in. And says, we offer a perspective on notable features of SARS-CoV-2 genome and discuss scenarios by which they could have arisen. Our analyses clearly show that SARS-CoV-2 is not a laboratory construct or a purposely manipulated virus. So that's what they came up with. In there, they did push out, and I'll just drag this up here and send this bring it to the front here. All right. They did put out here, though, they showed us here SARS-CoV-2 compared to the rat TG13, and they even noted that there's this big gap here where you have this PRRA sequence that somehow magically got inserted into SARS-CoV-2. Not a mutation, right? They even pull up, hey, pangolin, right? It's missing that sequence. Rat TG13 is missing this sequence here, right? Human SARS itself, classic SARS, missing this sequence here. Bat, SARS-CoV-related, missing this sequence here. So this is a really important finding. And because of this is a really important finding, mutations happen all the time, but when you have an insert, when you have a whole insert, that's kind of the thing that humans would do. They would put a whole insert in there, and you have to explain where that insert came from because whole inserts are not part of the mutation pathway. That's a whole different process of how things get inserted, all right? So if I'm the journalist, you ask this question and you ask about the PRRA and nothing else. So let's look at they only mentioned furin 4 times in this article by these world-leading experts on virology saying that this thing is completely natural. Well, the sudden appearance way out on a family tree, which has, and by the way, virologists all know this stuff, right? I had to go learn this stuff, but presumably they're all up the curve on this. To suddenly have this thing way out here and all alone on its family tree have this furin cleavage site, you'd think that would be important. Well, they only mentioned the word furin 4 times in this entire multi-page article here on the proximal origin of SARS-CoV-2, and here's what they finally said. They said the functional consequence of the polybasic cleavage site in SARS-CoV-2 is unknown. Wait a minute. What do you mean it's unknown? The functional consequence of a polybasic cleavage site is very, very well known, and I'm going to show you how well known it is. So that's dissembling. That's not true. The functional consequence of polybasic cleavage sites is it allows SARS-CoV-2 to be a lot deadlier and a lot more able to enter into the cells, okay? And it will be important to determine its impact on transmissibility and pathogenesis in animal models. So here's the weasel wording. They're just like, well, listen, we know that polybasic cleavage sites are really important in other viruses, but we haven't proven it yet in animal models, so we can't really say. So they say it's unknown. But if you asked a virologist what that would confer, I think they'd pretty much tell you what would happen. which is nothing good. Experiments with SARS-CoV, that's classic SARS, have shown that insertion of a furin cleavage site at the S1-S2 junction enhances cell-cell fusion without affecting viral entry. In addition, efficient cleavage of the MERS-CoV spike enables MERS-like coronaviruses from bats to infect human cells. Huh. Well, so I guess they're saying in classic SARS, somebody inserted a furin cleavage site at the S1-S2 junction, and it didn't really help all that much. But in MERS-CoV, it clearly enables coronaviruses from bats to infect human cells. All right, let's go here. Nah, I'm going to go here first. I'm going to come back to that. All right, so check this out. So have people been monkeying around with this? Yeah, so check this out. The closest relative of SARS-CoV-2 with a furin site is the HKU5 strain. This was isolated by who? Oh, Batwoman back in 2014 in Guangzhou from bats. But it is a very distant relative. Their spike proteins only share 36%. So the virologists are puzzled. Where did this 12 nucleotide insert come from? Could it be lab-made? Well, virologists have studied furin sites in coronaviruses for decades and have introduced many artificial ones in the lab. For example, an American team inserted RRSRR into the spike protein of the first SARS-CoV back in 2006. So they investigated whether proteolytic cleavage at the basic amino acid residues were to occur might facilitate cell-cell fusion entry. We mutated the wild-type SARS glycoprotein to construct a prototypic furin recognition site, RRRR, at either position. So, hey, Americans did this back in 2006, right? They inserted these things.
But your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should.
And again, in 2008, the Japanese inserted RRKR into SARS-CoV protein in 2008. And so they did that.
But your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should.
And again, in 2008, Dutch colleagues also studied how these protease sites of SARS-CoV and compared them to the murine coronavirus, which has a site like this, right? Which again is a polybasic site, quite similar to that of CoV-2. Look how close those are. SRRAHR, and this is SPRRAR, right? So very close. And in 2009, another American group also working on improving SARS-CoV, classic SARS-CoV, and continuing the American tradition of not penny pitching on arginines, they inserted as many as 4 of them, RRSRR. So have humans been putting these polybasic cleavage sites into viruses to study how those influence the viricity of these things, right? The ability for them to both transmit and then infect into the cells? Yes. It's a huge line of research that's been going on for a very, very long time.
But your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should.
And finally, again from this wonderful thing in Medium by Yuri Dagan here, in Beijing, 2019, but the most recent work of this kind that I came across, he writes, was an October 2019 paper, from several Beijing labs where the new furin site RRKR was inserted not just some pseudovirus, but into an actual live chicken coronavirus infectious bronchitis virus. There they are inserting it. So this kind of stuff has been going on for a long time. And it's that cleavage site right here that allows that SNP to happen so that we can get that second action happening so that this thing comes in and the membranes fuse, and that confers so much of the terrible, awful nature of this particular virus. So it was odd to me then to see on the Joe Rogan Experience on March 10th, one of the leading virologists in the United States, Michael Osterholm, came on and was asked by Joe, is there any chance this thing came out of a lab? And Michael gave an answer where he said, man-made diseases, that's exactly, we could not have crafted a virus like this to do what it's doing. We don't have the creative imagination or the skill set. Yeah, we do. We got the skill set. Yep. There it is. Yeah. Okay, so we've been monkeying around with this stuff for a long time. Michael knows this better than anybody. He's one of the leading virologists out there. So that's kind of dissembling on his part. And further, he said, well, and this thing, it comes from a pangolin probably. It's really close to a pangolin. And of course, Michael would know that it's close to a pangolin, but it's missing a polybasic furin cleavage site, PRRA. So if you're a journalist out there, this is the smoking gun. This is the number one thing you want to find out. Where did this insert come from? And you would be well to know that these sorts of inserts have been monkeyed with over and over and over again by scientists seeking to understand how viruses do what they do. And by the way, they have two-step process. For step 1, bind the receptor. Step 2, get proteolytically cleaved so that you can undergo a fusion process so your RNA as a virus gets dumped into the host cell so it can replicate. All right, and clearly, he says, we don't have the skill set. Clearly, we have these capabilities because here's a group. This just came out today, actually. Yep, May 4th. That says, oh, hey, that whole thing, the SARS-CoV-2, you know what they did? They used a synthetic genomics platform, and only a week after receipt of the synthetic DNA fragments, you know what they did? They were able to engineer and resurrect chemically synthesized clones of the recent epidemic SARS-CoV-2. The technical advance we describe here allows a rapid response to emerging viruses. I'm going to edit for them here, as it enables the generation and functional characterization of evolving RNA virus variants in real time during an outbreak. So they were able to, using an artificial yeast model, they were able to generate infectious RNA to rescue viable virus. They rebuilt the virus out of fragments. So to say, we just don't have the skill sets for this stuff, not true. Because we actually do have that. And remember, you look at this and you say, oh my God, there are people who are able to recreate this virus from scratch, in essence, just having the sequence. I'm not so sure I feel comfortable having that information out in the world. Because obviously it could be used by the wrong people in the wrong way. And remember...
Yeah, but your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should.
All right, and I think we're getting a real circling of the wagons here as well. In this one, in April 20th, we have scientists are tired of explaining why the COVID-19 virus was not made in a lab. This is in sciencealert.com. It's a rumor that just won't die, they write in an exasperated tone of voice here. When asked whether COVID-19 virus was genetically engineered in a lab, scientists have already said no, rather firmly, but the matter of the new coronavirus's origin is unlikely to be put to rest so easily. Back in late March, we covered a study published in Nature Medicine in which the researchers investigated the genomic data of SARS-CoV-2, particularly the receptor binding domain sections of the virus, to try and discover how it mutated into the virulent and deadly version we're currently struggling to contain. Well, that's easy. It's got a polybasic furin cleavage site added to it. That's how it's so bad. As a byproduct of the research, they were able to determine that SARS-CoV-2 was not genetically manipulated. Now, how could that be? How can you determine that? So you can't actually say that. You want to know why? Because you look at certain technologies like this, and there are people who are in labs that have created entire genomes of various viruses without any signatures, and like there's a digital thumbprint that you might be able to find, if you're doing computer forensics. Well, what you would want to see is there some specific sequence of nucleotides that if you saw that, it would say that, oh, they were using a certain plasmid or a certain technology, or there was a certain snip and tuck. Nope. They have fully 100% recreated. Some labs have been able to fully recreate viruses with no alterations, just 100% the genome that you see without any telltale fingerprints going on in there. So you cannot actually determine it was not genetically manipulated just by looking at the genome. So this is crap what they're saying right here, pure crap. By comparing the available genome sequence data for known coronavirus strains, we can firmly determine that SARS-CoV-2 originated through natural processes. One of the researchers, immunist Christian Anderson. Oh, yeah, there we go. So you write the paper that proves that proves that this thing came from a natural origin, and then later you get quoted and you say, you quote yourself, I guess. But here's the thing. Comparing available genome sequence data to that of known coronavirus strains, how can you firmly determine this came through natural processes? Because when we do that, when we actually look at the genomic data, what do we find? We find that there's SARS-CoV-2 all by itself, way out here, all on it in its own family tree, with nothing that we can show for it here to show how, how, I don't even understand how that virologist can say that definitively and say, looking at known genomes, we can confidently say this thing's natural origin. Really? Where did it get that polybasic furin cleavage site from? Not from its neighbors, not from any of its closest relatives. So that's a little bit of a hop, skip, and a jump, don't you think? At least enough where you have to soften that and you can't be this declarative. It's this level of declaration here. By comparing available genome sequence data for known coronavirus strains, we can firmly determine that SARS-CoV-2 originated through natural processes. I'm not sure how you do that, because you can't look at the genome sequence data I've got and say anything except, wow, it's really weird that this thing somehow came up with its own furin cleavage site way over here all on its own. Two features of the virus, the mutations in the receptor binding domain portion of the spike protein and its distinct backbone, rules out laboratory manipulation as potential origin for SARS-CoV-2. This is the whole, it's too complex for a computer to have created it, so it can't have been done. And then they continue on. Look at this declarative language here. Although it is clear, now that we've established that the virus was not created in the lab, there have been ongoing concerns that may have escaped a research facility, with most of the speculation understandably focused on the Wuhan Institute of Virology. However, just remains speculation. The Washington Post recently reported the U.S. Embassy officials had safety concerns about the lab back in 2018, and the Institute did keep a closely related bat virus, but even that's far from a smoking gun. No, you know what the smoking gun is? PRRA. It's that polybasic furin cleavage site, which somehow came out of nowhere on the family tree and was just parked in there. And oh, by the way, just happens to be the place that all these gain-of-function virologists monkey with all the time, because it's a well-known fact for both many of the influenza viruses as well as the coronaviruses that you need to have that site. And if you can put that site in just the right spot in the spike protein, especially the S2, that you give, and you confer massive gain of function on that particular virus. So I would say, even though I'm not a world-leading virologist, that I can't look at this wheel and say that there's any case to be made for the genetic sequence of this supporting the idea that it has a close relative that we can look at where it got that polybasic furin cleavage site. And again, this is an insert, not a mutation. If it was a simple case of, 2 or 3 mutations and we got here and this one was close, all right, I could buy that. We don't have that. We've got a giant insert in here that gives this thing its gain of function. That's really, really bad ass. All right. Conclusions for today. The press needs to ask one question only. Don't be distracted by all of this complicated, we've run huge computational simulations. Oh my gosh, our genetic blah, blah, blah. No, here's your question. You ready for this, journalist? How did that polybasic furin cleavage site, PRRA, get into COVID-19? Full stop. As soon as you answer that effectively, we can move on to the next set of questions I've got, but that's it. Let's just start, if we could just focus on that. How did that cleavage site get into COVID-19, right? That's the important question. And I'm super disappointed with the dissembling, and I think what even amounts to overt lying and cover your A behavior, CYA behavior of what appears to be a significant portion of the U.S. virology community. And just to yeah, I skipped over. Let me go back to this. This is Francis Collins, director of National Institute of Health, right? March 26th. Look at this stuff. No matter where you go online these days, there's bound to be discussion of coronavirus disease. Some folks are even making outrageous claims that the new coronavirus causing the pandemic was engineered in a lab and deliberately released to make people sick. Well, see, he made a jump there I wouldn't have made. I don't know if it was deliberately released or not. Beats me. So, let's not conflate those two things. So I think that's some sophistry right there. It's like, oh, you believe it was engineered, then you also must believe it was deliberately released. No, back up. Separate concepts. This one requires speculation as to intent. This one, engineered, just requires us to look at the available data and ask some questions from which we can deduce certain things. So let's not put those two together. A new study debunks such claims by providing scientific evidence that this novel coronavirus arose naturally. Oh, I wonder what study that could be. The reassuring findings are the result of genomic analysis conducted by an international research team, partly supported by NIH. Oh, there's the conflict right there. In their study in the journal Nature Medicine, Christian Anderson, Scripps Research Institute of La Jolla, California, Robert Gary, and their colleagues used sophisticated bioinformatic tools to compare publicly available genomic data from several coronaviruses, including the new one that causes COVID-19. Certainly sounds very authoritative. The researchers began by homing in on the parts of the coronavirus genomes that encode the spike proteins that give this family of viruses their distinctive crown-like appearance. All coronaviruses rely on these spike proteins to infect other cells, but blah, 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 blah. Look at this. So here's the director of the NIH coming out and citing this same study here, the one that just skipped right over PRRA, gave it a little bit of short shrift over here, and then just wandered off into its conclusion territory, which was, this thing is guaranteed came from natural place, came from a natural setting. It was not purposely manipulated. You can't actually deduce that from the genomic sequence because, again, there are ways to take the genomic sequence of viruses and completely manufacture them from scratch and not leave a single fingerprint. So you can't actually look at the genetic sequence and say squat about it, as far as I understand it. All right. So this dissembling, overt lying CYA behavior, of these people, I would feel better if these virologists in the United States would say something more honest, like, hey, this looks really bad. I'm a little bit worried. I got to be honest. My whole career, my reputation, my paycheck are all kind of dependent on this research continuing. And listen, here's what we know for sure, and here's what we don't know for sure. Listen, we can't ever say for sure whether something has been monkeyed with or not, because the way the technology has progressed at this point in time, you could recreate an entire virus from scratch and not leave a single fingerprint. So we can't be sure based on looking at the sequence as well. Secondarily, when we look at this, we admit there's a big jump there, how this got this 12 nucleotide insert in there, which conferred magically a polybasic furin cleavage site. That's a mystery too. What would really be best is if we could go out and find a natural virus that actually mirrors this one perfectly, or close enough that we could say we got there. And by the way, everything that's kind of close, the rat TG13, the pangolin, everything we can find that's sort of close, none of them have that furin cleavage site. The kinds we've been monkeying with as gain-of-function researchers all these years. I know it looks bad, but here's why I happen to think that this is actually natural, right? Okay, that would be great. So I don't want to cast aspersions on the entire virology community. I'm saying at least those who have been monkeying with gain-of-function research, they seem to be the ones who are most interested in coming out and muddying the waters right now. And by the way, both the U.S. and China, up to their virologist eyeballs in this story. And again, not virologists, the gain-of-function virologist eyeballs. Let's just be clear. That seems to be a fairly small club that were involved in this stuff at this point. And by the way, these sorts of studies should never have been done. The things we could have learned from them was tiny compared to the amount of damage that could be done by this. And if this turns out to have been a gain-of-function research experiment that went bad, whether it was released deliberately, accidentally, unintentionally, intentionally, it doesn't matter. It happened. And if that's the case and that's the story, there's a lot that has to be answered for, because this has been one of the most destructive economic events of anybody's lifetime. And that should have been a foreseen consequence. Somebody should have been asking that question. Hey, what if one of these things got out? And the fact that they didn't ask that question or answer it appropriately, and meaning they still decide to go ahead with it, however you look at it, this is now out of the bag. So that's as far as I'm going to go with this story, I think. I think I don't have to go much further than that. To me, this is the one thing I would focus on. I wouldn't get PRRA. I wouldn't get too wrapped up in anything else about this at this point in time. And until that question is answered, I'm going to have to fall very heavily on the side of believing that this was an intentional insertion of a polybasic furin cleavage site in order to explore and unravel the mysteries of how a virus penetrates a cell. I think it was a gain-of-function research experiment that was both successful and then unsuccessful because of what happened next. So that's all I've got on that for today. Remember, plant a garden. I just don't think we're getting out of the woods on this particular virus anytime soon. It's got a lot of tricks up its sleeve, and the problem is that particular furin cleavage site that it has, one of the reasons it's so bad, so badass, is because there are both intracellular, meaning inside the cell, and extracellular, out in the bloodstream and in the lymphatic tissues. There are proteases existing both inside and outside of cells that can help activate this virus. So that means that it's really, really particularly good at infecting people and getting inside our cells and doing a lot of damage. And by the way, with both ACE2 and CD147 receptors, it has a lot of different places that it can attack inside the body. That's what makes it particularly bad. And that's why we shouldn't have been monkeying around with these sorts of viruses in this particular way. Very bad idea. And unfortunately, we're going to have to live with the consequences of that if the story as I've laid it out turns out to be true. All right. Thank you for listening. And we'll be back tomorrow. Bye.